Hello dear students, welcome back again to Chinmoyee's biology channel. So today I am here with the second part of life processes. So first part, I think you have all gone through the first part where I have discussed about the autotrophic nutrition in details. So today I have started with one activity of autotrophic nutrition which was left previously, and with heterotrophic nutrition, nutrition in organisms, and the full process of digestion, that is nutrition in human beings. So how from mouth to anus the full digestive system works, that we are going to discuss today. So without further delay, let's get started with today's brand new episode of Life Processes. So hence today we are going to discuss about this activity 5.2. So last day we have already discussed about photosynthesis, about how does a stomata look like, about the guard cells and all. So here what we are doing, we are taking two potted plants like A and B we are taking, and we are placing them in a dark room for 3 days, and now we are placing each plant on separate glass plates, and we are taking one watch glass containing potassium hydroxide by the side of any one of the plants. Now what is the function of potassium hydroxide is that it is absorbing carbon dioxide. So its function is to absorb CO2 or carbon dioxide. Okay, now we are covering both the plants with separate bell jars as shown in this diagram, and you are using Vaseline to seal the base. So we are Vaseline used here to seal the base. Now keep the plants in sunlight for about 2 hours. Now pluck a leaf from each plant and check for the presence of starch as in the above activity. So what do you see? Do you see the leaves show the presence of the same amount of starch? What do you conclude from this activity? So what conclusion we are getting? First of all, as there is no carbon dioxide present, no photosynthesis happens here. So here no photosynthesis. Okay, as one of the raw materials is carbon dioxide, so no photosynthesis happens in this part. So as no photosynthesis is happening in this part, so the leaves of this plant will not have any starch in them. Absence of starch in the leaves. Whereas in B, as there is no potassium hydroxide, carbon dioxide is there, and photosynthesis happens in this plant, and there is production of starch in the leaves. So as the leaves are having starch in this B plant, we can say that presence of starch in B plant and absence of starch in A. So this is the conclusion. Now based on the above two activities performed above, we can design an experiment to demonstrate that sunlight is also essential for photosynthesis. So like in this experiment, we have concluded that carbon dioxide is essential for photosynthesis. We can also design an experiment to prove sunlight is essential for photosynthesis. So how you can set the experiment? We can keep one potted plant in darkness for a few days, and one potted plant in sunlight. So if we are plucking leaves from both the plants, we will see that the plant in dark will not have any starch, whereas the plant kept in sunlight will have starch. So photosynthesis can happen only in presence of sunlight. So there like this it can be proved. Now, so far we have talked about how autotrophs meet their energy requirements, but they also need raw materials for building their body, right? So water used in photosynthesis is taken up from the soil by the terrestrial plants. Other materials like nitrogen, phosphorus, iron and magnesium are taken up from the soil. Nitrogen is an essential element used in the synthesis of proteins and other compounds. Now this is taken up in the form of inorganic matters or nitrites. So we have told that nitrogen is taken from the roots in the form of nitrates or nitrites, as the atmospheric nitrogen is converted into nitrates or nitrites by the nitrogen fixing bacteria. So nitrogen fixing bacteria is there. So nitrogen fixing bacteria like rhizobium and all, they are helping in the fixing of the atmospheric nitrogen to nitrates or nitrites. Okay, so example may also be given. So example, we can say that example is also given, that is rhizobium. It is a nitrogen fixing bacteria. Now, or it can be taken up as organic compounds which have to be prepared by bacteria from atmospheric nitrogen. So, from atmospheric nitrogen it is converted. So, now next we will see What is heterotrophic nutrition? Now, heterotrophic nutrition, each organism is adapted to its environment. To form a nutrition, it differs depending on the type and availability of the food material, as is how it is obtained by the organism. Okay, so for example, whether the food source is stationary, such as grass, or mobile. So the food source may be stationary, such as a grass, or it may be mobile, such as deer, would allow the differences in how the food is accessed and what is the nutritive apparatus used by a cow or a lion. So there is a range of strategies by which the food is taken. So it depends on whether you are taking a food which is mobile or it is stationary. So there are a range of strategies in which the food is taken. So some organisms, they break down the food material outside the body, where before they absorb it, then they are absorbing it after they are breaking it outside the body. So examples are fungi like bread molds, yeast, mushrooms and others. They take whole material, break it down inside their bodies. What can others be taken in and broken down depends on the body design and functioning. So some other organisms derive their nutrition from plants or animals without killing them. So this is that they are retaining or deriving their nutrition from plants or animals without killing them. So they are also taking their nutrition. So this type of nutrition is called parasitic nutrition. So this strategy is used by a wide variety of organisms like cuscuta or amarbale, ticks, lice, leeches, tapeworms. So this type of nutrition is called parasitic nutrition. Okay, so this is called parasitic nutrition. Now, how do organisms obtain their nutrition? So since the food and the way it is obtained differ, so the digestive system of different organisms also is different. So the food is different, the form of the body is also different. So some are single-celled animals or organisms, some are multicellular organisms. Is it clear to you all? So since the food is different, the way of consumption of food is different, the digestive system is also different. Now in single-celled organism, the food may be taken in by the entire surface. Okay, so as complexity of the organism increases, different parts become specialized to perform different functions. So when it is a unicellular organism, the full body, entire surface can take the food. But when different parts are there, when it is a multicellular organism, then each part is having a different function. Like in our case also we can say that we can tell that mouth is having is a part which is engulfing the food. There are other organs which are digesting the food, other organs which are absorbing the food, and then they are excreted through one part of the body. So we are multicellular organisms, so it is a complex process. But in unicellular organism, it is very simple. Now only the full entire surface area is taking the food, there only it is digested, there only it is excreted. So that is the way the body is adapting itself. So let us take an example of amoeba. It takes its food using a temporary finger-like extension. So this is amoeba, this is the food particle. So these are the temporary finger-like projections which are called pseudopodia. Okay. So these are finger-like extensions of the cell surface only which fuse over the food particle. See, they are fusing over this food particle and they are creating a food vacuole. Inside the food vacuole, the complex substances are broken down. Inside they are broken down into simpler ones. When then they are diffusing into the cytoplasm. Okay, so they are diffusing into the cytoplasm once they are broken down into simpler form. That is, the digestion is happening into their body and the food is being absorbed. So food particle, it's being engulfed, food vacuole is prepared and it is slowly digested into simpler form into the cytoplasm. So the remaining undigested food material is moved onto the surface of the cell and is thrown out. So the undigested part is moved on the surface of the cell and it's immediately thrown out of the body. So no need of excretory system, digestive system. So ingestion, digestion, assimilation, absorption, all these are complex processes in multicellular organisms or humans or animals who are having a complex organ system. Is it clear to you all? So these are simple process organisms. They are having simple processes, how they are taking, engulfing, digesting, and how the food gets mixed up or diffused in cytoplasm. So the remaining undigested foods is moved to the surface of the cell and is thrown out. So what is happening in case of paramecium? Same thing, which is a unicellular organism. The cell has a definite shape and the food is taken in a specific spot. Food is moved to this spot by the movement of the cilia. So pseudopodia, if it is a unicellular organism like this, full body is having cilia. So if they are seeing a food particle here, what will the cilia do? As it is staying in water, as it is a water animal, so with the help of the cilia, the food particle is being engulfed inside. Food is moved to the spot and they cover the entire surface of the cell. So it is also through this only it enters the body. The entire cell covers the food, and in the same way as in amoeba, it is being digested in the body. So this is all about the digestion in organisms. Now we will move on in the next slide about the nutrition in human beings. Is it clear? Nutrition in human beings. The alimentary canal is basically a long tube extending from the mouth to the anus. Now, in the figure down, we will see that how the alimentary canal starts and where it ends. So, we can see the tube has different parts. Various regions are specialized to perform different functions within this alimentary canal. So, what happens to the food once it enters the body? We shall discuss this full process here only. Okay. So, first let us take one activity. Now, take a 1 ml starch solution in 2 test tubes A and B. Suppose these are 2 test tubes A and B. We are taking 1 ml starch solution in each. Okay, so 1 ml starch solution in each test tube we are taking. Now add 1 ml of saliva in test tube A. So here we are adding saliva and leaving the both test tubes undisturbed for 20 to 30 minutes. Okay, so we are leaving both the test tubes undisturbed. Now adding a few drops of iodine solution to the test tube. So we are adding few drops of iodine solution. We are adding few drops of iodine solution to both the test tubes. So in which test tube do you observe a color change? What does this indicate about the presence or absence of starch in the 2 test tubes? So here both the test tubes are having starch only solution 1 ml, but in the first test tube we have added few drops of 1 ml of saliva, and then we have added few drops of iodine solution in both the test tubes. So what does this tell us about the action of saliva on starch? So this answers in detail I'll be discussing in the next slide. So let us explain the answer first. So here, what is the solution? So starch is a component in many of the foods. So when iodine is added to starch, it turns blue, or we can say that blue-black color. So that is why in the experiment the solutions of both the beakers turn blue, because both the beakers are having starch, so both of them will turn blue first. So in addition to saliva, which contains an amylase enzyme, a starch digesting enzyme, chemical compound that catalyzes the breakdown of the starch. So the starch is gradually converted to sugar. So what is the starch being done? The starch on action of amylase is gradually converted to sugar. Okay. So thus once the starch is broken down, the blue color disappears. So first it was blue, and then when saliva was added, then this color changes to colorless. So it disappears. Okay, the color disappears. Now it is important to know that salivary amylase requires an alkaline medium to act. So this is the solution of what the questions we have asked in the previous experiment. So it always requires salivary amylase, it requires always an alkaline medium to act. So now we will go to details about the entire digestive system in humans. Okay. Your answer to the previous question, I think you have already got a clear answer to this question in the previous slide, that what does this indicate that about the presence or absence of starch in those test tubes, and what does this tell about the action of saliva on starch. So you got the answers, right? So now we will move on to the full digestive tract and what is happening in this digestive tract from mouth to anus. Okay. So we are eating various types of food which has to pass through the same digestive tract. So we know we are eating it through the mouth and constantly through the esophagus, through the whole digestive tract. So this is the digestive tract. So first it is entering, this is tongue. This is the esophagus, this is the mouth, and through that it's coming down. This is the stomach, then after the stomach is the small intestine, then it comes to large intestine, and then it is anus. Okay, so here the pancreas, the liver, they all are there. So liver and pancreas is there, the bile duct, it's here, gallbladder is here. So all are here, and this is the full digestive tract. Okay, so you should know the labeling of the digestive tract, and this full diagram with labeling I'll be giving in the next slide, that the full detailed diagram with labeling should be given in your exams, right? So might be they'll be giving you 1 or 2 labelings, or they can give you the diagram and pinpoint to label 1 or 3 or like that. So it's you should know the detailed diagram of digestive tract with labeling. Okay, so naturally the food has to be processed to generate particles which are small and of the same texture. So this is achieved by crushing the food in our teeth. First we are crushing the food in our teeth. Since the lining of the canal is soft, we should make the food soft and also wet it to make its passage to the passage smooth. So once we can't take something hard and just engulf it. No, it's not like that. It should be chewed, it should be masticated, it should be made smooth to pass through the esophagus. When we eat something, we like our mouth waters. So this is actually not only water but a fluid called saliva. So this is saliva secreted by the salivary glands. So salivary glands are present here near the mouth only. So these salivary glands are secreting saliva. So another aspect of food we ingest is its complex nature. It has to be absorbed by the alimentary canal and it has to be broken down into smaller molecules. So how it is happening? So there are catalysts in our biological system which are called enzymes. So biological catalysts are what? They are enzymes. So the saliva contains the enzyme called salivary amylase. So saliva contains the enzyme called salivary amylase, and that breaks down the starch, which is a complex molecule, to a simple sugar. So starch, with this salivary amylase, the starch is converted to simple sugar. And then the food is mixed thoroughly with saliva and moved around the mouth while chewing by the muscular tongue. Now the tongue is chewing with the help of teeth, with the help of saliva, with the help of tongue. It is made smooth and then it is swallowed gradually through the esophagus, and it is necessary to move in a regulated manner along the digestive tube so that it can be processed properly in each part. So the lining of the canal has muscles that contract rhythmically in order to push the food forward. So these esophagus, there are muscles in that which are causing the peristaltic movement in the gut. So in the next slide I will be discussing with the detailed diagram how this peristaltic movement is happening when we are taking a bolus of food. So from the mouth, the food is taken to the stomach through the food pipe called the esophagus, and the stomach is a large organ which expands when the food enters in it. The muscular walls of the stomach help in mixing the food thoroughly with more digestive juices. Now we will put a stress on this peristaltic movement in the next slide, and then we will come up to the stomach part. Here in this slide, we are seeing two diagrams. First one is the diagram of the full digestive system. It is a very important diagram. You should learn this full with the labeled parts which have been given here. These are salivary glands, esophagus, stomach, mouth, liver, gallbladder. All this labeling should be memorized. Pancreas, and this is a very clear picture for you. I have taken out this picture because it is very easy and very simple to draw this picture. Small intestine, large intestine, appendix, rectum and anus. Okay, so this is the important diagram of digestive system. You should learn, you should memorize and you should practice it. Now this is the peristaltic movement which I told I'll be discussing in detail. So what is this peristaltic movement? So this peristaltic movement or peristalsis we can say. So this movement is actually when you are eating a bolus of food, the muscles here, once they are contracting and once they are relaxing. Once they are, these circular muscles contract, constricting the passageway and they are pushing down the bolus. And then again this muscle is relaxed and the bolus of food is pushed down. Now there is gastroesophageal sphincter over here. So what this sphincter is doing, this is also this gastroesophageal sphincter. When this sphincter is open, then this bolus of food will enter the stomach. And until and unless this is open, the food will stay in this esophagus itself. So there is a contraction and relaxation movement. So alternate contraction and relaxation movement of the muscles of the esophagus happens when we take a bolus of food, when it is chewed, when it is masticated in your mouth and when it is swallowed and when it moves down the esophagus. So once contraction of the muscle happens and once relaxation, and then the sphincter opens, and once the sphincter is open, gastroesophageal sphincter is open, then only food enters and then again it gets closed. Why it is closed again? So that the food bolus can't move back up this esophagus again. So no reflux should be there. Okay, so in some people you have seen they are having chest pain, burning, heartburn, and also why this is having, because excessive acid is secreted in the stomach and this sphincter is not closing on time. So the reflux of this food happens in this esophagus. So then they are having as the gastric esophageal sphincter is not closed properly, they are having GERD, or that syndrome is called gastric esophageal reflux disorder. So this esophagus, the gastric disease which is causing the esophageal reflux, so that is gastric esophageal reflux disorder. Now we will move on with the processes which are happening in the stomach. Let us move on. Now, through that peristaltic movement from the mouth, the food is taken to the stomach, and the food pipe or through the food pipe or the esophagus. So, in the stomach, there are several digestive juices. The digestion in stomach is taken care by several gastric glands. So, why the gastric glands are secreting the gastric juices? Okay. So, that helps in the digestion of the food material in the stomach. Now, these gastric glands are present on the walls of the stomach, and these release hydrochloric acid and a protein digestive enzyme called pepsin and mucus. So, the hydrochloric acid creates an acidic medium which facilitates the action of enzyme pepsin. So, as the hydrochloric acid is secreted, it facilitates the environment for the enzyme action, for the action of the enzyme pepsin. Okay, so this action of this pepsin is facilitated by hydrochloric acid. Now, what other function do you think is served by the acid? The mucus protects the inner lining of the stomach. So this stomach is having an inner lining, so that inner lining is protected by the mucus from the action of the acid under normal circumstances. Now, we have often heard of adults complaining about acidity. So, can this be related to what is discussed above? Yes, obviously. When we can say at times when we stay empty stomach for a long period of time, gradually what happens? The mucus layer gets corroded away by the concentrated hydrochloric acid, and there ulcers happen. So, this causes gastric ulcers, and that causes at times that can cause acidity, because the environment has become too acidic. Okay, so it's what we can say, that it's excessive acid, and the mucous membrane, as it has got corroded away, so directly the acid is falling on the stomach wall and it's getting ulcerated. So gastric ulcers happen in this way. Then comes the exit of the food from the stomach is regulated by a sphincter muscle. So how the entry was related with the help of a sphincter muscle, we have seen that gastroesophageal sphincter muscle was there. Similarly, when the food exists from the stomach also, there is a sphincter muscles. So that allows the food from the esophagus to the small intestine, from the stomach to the small intestine. So small amounts of food enter the small intestine from the stomach, and from the stomach the food now enters the small intestine, and this is the longest part of alimentary canal. So in human beings it is very long, and it is fitted into a compact space because of extensive coiling. So in our stomach the small intestine is largely coiled, and it is fitted in a compact place. The length of the small intestine differs in various animals depending on the food they eat. So herbivores, they are eating grass, they need a longer small intestine because they are allowing to digest the cellulose. So cellulose takes longer time to get digested, so they are having a longer small intestine, whereas meat is easily digested. Hence, the carnivores like tigers have a shorter small intestine. Is it clear? So, tigers are having shorter small intestine. So, small intestine is the site. What is the site important for? Where the complete digestion of carbohydrates, proteins and fats are taking place. It receives the secretions from the liver, pancreas for this purpose. So, it is receiving the secretion of the liver and the pancreas. What are the secretion of the liver and pancreas? So these secretions, the liver is secreting bile and the pancreas is secreting insulin and glucagon. So these are being all put together in the small intestine for digesting of the food substances. That is all the carbohydrates, proteins and fats. The food coming from the stomach is acidic and has to be made alkaline for the pancreatic enzymes to act. So the pancreatic enzymes, they can only act in the alkaline environment. So bile juice, which is coming from the liver, it accomplishes this in addition to acting on fats. Fats are also present in the intestine in the form of large globules, which makes it difficult for the enzymes to act on them. So in the intestine, fats are present in the form of large globules, and they are making the enzymes difficult to act on them. So what is the action of these bile salts? They break down them into smaller globules, increasing the efficiency of the enzyme action. So bile salts are breaking all these fat globules into the simpler ones, smaller globules, increasing the efficiency of enzyme action. So this is similar to the emulsifying action of soaps on dirt. So in the chapter 4 you have already studied, in the previous chapter only you have already studied the emulsifying action of soaps on dirt. So like that only, fat globules are being broken down by this bile into a simpler smaller globules, and which increases the efficiency of the enzyme action. Now till now I think it is clear. So if you are having till any doubts, please do write in the comment box so that I can take that topic and discuss in the next episode. Now the pancreas secrete the pancreatic juice, which contains trypsin. So pancreatic juice is containing trypsin, which is digesting proteins, and lipase for the breaking down of emulsifying fat. So trypsin is digesting the protein, whereas lipase is acting on the fats. Okay. So trypsin is acting on the proteins, it is breaking down the proteins, lipase is breaking down the fats. The wall of the small intestine, they are having several ducts which are secreting the intestinal juice. Okay, the enzymes present in it finally convert the proteins to amino acids and the complex carbohydrates into glucose, fats and fatty acids and glycerols. Okay, so complex carbohydrates are converted to glucose and fats into fatty acids and glycerols. So it's very simple. So different types of enzymes are acting on different types of food substances and breaking them down into smaller parts. Trypsin is acting on proteins, lipase is acting on emulsified fat. Enzymes present, finally they are converting proteins to amino acids and the complex carbohydrates into glucose and fats into fatty acids and glycerol. Now this is all about the small intestine. Now digested food is taken up by the walls of the intestine. The inner lining of the small intestine has numerous finger-like projections called the villi. So these villi, they increase the surface area and then absorption and assimilation takes place. And the villi are richly supplied with the blood vessels which take the absorbed food to each and every cell of the body. So if the absorption is happening properly here, then all the proteins, carbohydrates, fats which are being absorbed in the small intestine will be taken to different parts of the body. So absorption is happening with the help of villi and they are rich in blood vessels, and these blood vessels are carrying all the nutrients which are being absorbed to different parts of the body, all cells to which are helping them or utilizing them to obtain energy, building up new tissues and repairing the old tissues. So this process will be well done in this way. Now the unabsorbed food is sent into the large intestine. So what is it is doing? Its wall absorb more water from this material and the rest material is removed from the body via the anus. So the exit of the waste material is regulated by the anal sphincter. So there anus also you are having a sphincter. So entry of the stomach also is having a gastroesophageal sphincter. Exit from the stomach to the small intestine is also regulated by a sphincter. And again here the excretion time, there is an anal sphincter. So all the entry and the exit points, they are all supplied with sphincter. So this is all about the digestive system from the mouth to the anus, including the small intestine, which is the site of complete digestion of carbohydrates, proteins, fats, and whereas there are villi which are absorbing all the nutrients and they are rich in blood supply, so they are all transporting the absorbed material to each and every cell which gives us energy. Okay. Now here is a note where we can say that what are dental caries? Dental caries are tooth decays which are caused by gradual softening of the enamel and dentine. Now it begins when the bacteria acting on sugars, they produce acids and soften and demineralizes the enamel and masses of bacterial cells. So bacterial growth happens together with the food particles which stick to the teeth. So it is very important that you should brush your teeth properly to clean the plaque and to form the dental plaque. Okay, so saliva cannot reach the tooth surface to neutralize the acid as plaque covers the teeth. So brushing the teeth after eating removes the plaque before the bacteria produces acid. And if untreated, these microorganisms may invade the pulp, causing inflammation and infection. And if this infection is too much, then tooth removal also can be done. So you should be aware of this dental caries and you should protect your teeth, brush your teeth properly so that no microorganisms can grow in your teeth. And it should be brushed properly so that saliva can reach the teeth to neutralize the effect of acid. As the plaque covers the teeth, so the plaque should be removed and saliva should reach to the tooth surface to neutralize the surface from acids. Okay, so these are dental caries. Now we will move on to a few questions and then the next part we will be doing in the next episode. Already we have dragged it so long, so it will be difficult for you to understand. So these questions also, after this full lesson, you just write this questions in your answer book and try to answer this as I have discussed everything. So what are the differences between autotrophic nutrition and heterotrophic nutrition? Where do plants get each raw materials required for photosynthesis? What is the role of acid in our stomach? What is the function of digestive enzymes? So take each and every enzyme and write for what they act, like trypsin, lipase, so all the enzymes, salivary amylase and all enzymes. You take each and every enzyme, whatever I have discussed above, write that only, you tell their function. So what is their role in the digestive system? How is the small intestine designed to absorb the digested food? So here what you are going to tell, you are going to tell about the villi, right? So what are the structures which are helping you to absorb the digested food? So about the structures you should tell. So you should discuss about villi, which are present on the surface area of the small intestine, which helps to absorb the food. And they are supplied with a rich number of blood vessels, which help to absorb the nutrients from the food, and they supply it to each and every cell, and which gives you energy in turn. So I think all the questions you can answer all the questions. If till now you are having any doubt, please do write in my comment box. And today this episode is completed. And in the next episode, very soon, we will be coming back with respiration, another very important life process. So till then, subscribe to my channel, stay tuned, stay updated with all the videos which I am posting. And if you are liking my channel, please do share it with your friends. And please don't forget to subscribe, as your subscription only helps me to get motivated to make more content, and it should be a better content, and I think it will help you a lot in your studies. So this episode is completed. So seeing you soon with the next episode in respiration.